welcome to this Antibiotics in Societies panel. This is our third panel in the series, and I'm delighted to have another excellent panel of speakers in today's seminar. Hopefully, I can still see people joining, but we'll make a move. So today, we're going to be talking about antibiotics beyond humans, ecologies, production and flows. And this is really timely because this week is World Antibiotic Awareness Week, just started today, and that is making us aware of antibiotics and other antimicrobials and where they are. They're around everywhere. And I think this panel will really bring that kind of to the fore as well, that we may, that antibiotics are in places that we may not usually think about. So just to overview for people who haven't been participating in our other panels so far, the objective of this panel series is to bring together key insights from social research studies that are looking at antibiotic use. And we're in particular wanting to look at how we can respond to the findings of social science about antibiotic use. What should we do about it? And so we are looking at antibiotics in health facilities. We've looked at antibiotics beyond health facilities, also primarily for human use. And today we're looking beyond human use of antibiotics. And in our next session, we'll be looking at science and technology and infrastructures. On our panel today, I'm delighted that we've got four excellent speakers. Steve Hinchliffe, followed by Klaas Kjelle, followed by Salas Ariola and Rijul Kochhar. And then we'll have a roundtable with the panelists, followed by some Q&A from the audience. So you'll see that you've got a Q&A function that you can type in your questions. We're going to have the speakers back to back, and they're doing 12 minutes each. And then we will then open up for discussion afterwards. Just to say that the webinar is being recorded and will be available on our website afterwards. And if you're having any technical issues, you can use the chat function. But for the questions that you're posing, please put them in the Q&A. This enables everybody to see them. Linked to the panel series, the AMIS team are collating a report. And this is going to put forward the implications that we're pulling together from across this panel series. And we're hoping to present this kind of cutting edge picture of research and what we should do about it in relation to antibiotic use. If you've been doing research on antibiotic use and want to contribute your findings to the report or the implications of your findings, we'd be really grateful to hear about it. And this is a link to a survey which we will send out to you. There's a poll that's just popping up. Is there? There it is. So you might see on your screen, can everybody see there's a poll that says, are you happy to receive follow-up email communication? This allows us to then send you the link to today's recording and to say thank you for attending, and also to send you the link to this survey. We won't send you anything else. Oh, look at that. Okay, brilliant. Thank you very much. Okay, so without further ado, I'm going to invite Steve to give our first talk. I'm stopping sharing. And you're on mute, Steve.
Okay, thank you, Claire. First mistake. Hopefully you can see my slides now, and I'll set off. So I've given a slightly provocative title of On Users, Why Social Science Might Contribute to the Wrong Outcomes. And I don't have the answers. This is a kind of plea for help as much as anything else, or a contribution to thinking about things. So I thought I'd start with a picture of a man in a bow tie, because I know Klaus usually does that. The historians usually do that, start with a picture of a man in a bow tie. But this is James Dusenberry, who was an economist, famous actually for distinguishing between social consumption and individual consumption, arguing that we tend to consume in groups rather than as individuals. But I really like this quote, and I stole it from Michel Callon and John Law's paper from some years ago. Economics is all about how people make choices. Sociology is all about how they don't have any choices to make. I quite like that because I think it captures what state we're at the moment in AMU. We've sort of been forced perhaps beyond our liking, many of us, to think about individual choice or choices as the key issue for antibiotic use. And now there's a raft of work talking about, well, actually people don't have much choice, all right? They've got those structural inequalities, all those kind of things are being emphasized. So that's one of the key areas, and neither really seems that satisfactory in the current situation. They both end up in places that we don't want to go. The voluntarism, I guess, of individual choice, and then the kind of structural glue of the kinds of things that we could argue that limit that choice. So what alternatives are there? And I suppose one way of doing this, and familiar to those who've been doing relational social science, and I suppose many of us would probably agree this is right, but what difference does it make? So what if actions about the environments, the material social ecologies then within which it unfolds, which I think most of us would probably have agreed with 10 years ago when we were doing field work that seemed to have less importance in some ways of actually doing something. So what difference does it make? And that's the difficulty I suppose I've got in some of this. But I want to give some examples of why it's important to think like this, and then maybe we could have a debate on why it's difficult to kind of move the questions on for future research agenda. So I've got two stories. One story, the paradox of disease burdens. So this focuses on work I did with Andrea Butcher, who will be coming up later as part of Sala's work as well, on Bangladeshi aquaculture. And one of the key issues here was the attempt to move farming of shrimp in this case and prawn from a highly diseased situation to one where there was a lower disease burden, and one of the knock-on effects would hopefully be that farmers would use less treatments. So NGOs like World Fish, but others, and the Ministry for Fisheries really started to push and put a lot of investment into disease-free hatchery seed, which would go out to farms, and then farmers would hopefully have lower disease burdens and therefore use less antibiotics or antimicrobials and other treatments. The adoption of disease-free technology required, though, change in farming and social practice. No good having disease-free seed. As soon as you put it in a pond, it was full of disease. So the idea, of course, is that like most technologies, it came with a baggage associated with changing, modernizing practice and so on. So just to cut a very long story short, analyzing a lot of farms, over 300 farms, in fact, looking at farm outcomes and looking at all sorts of kind of measures, and surveying and doing interviews with those farms, spending time with the farmers, we demonstrated that disease burden is managed for most farmers by spreading risk. Now, I don't mean spreading as in spreading transmission, but actually spreading it like you'd spread bets. So it's about having multiple crops, stocking frequently, so maybe after a monsoon you put more seed in because the water salinity's changed and so on. All sorts of kinds of rules of thumb we use, mixing species, seed sources, harvesting opportunistically to take advantage of increases in price and those kinds of things. This was about spreading risk in the sense of ensuring yourself against collapse of a crop. So to realize the benefit of new seed, though, nonetheless, the farmers, the NGOs and others argued, and the hatcheries argued, they needed to adopt single or batch stocking and harvesting. Now, to get a sense of risk, I won't go into any of this, but one thing is very obvious in most farms is that you're in a kind of value chain where the farmers actually shoulder the burden of risk, okay? So the processor, exporter can choose, and there's about 200,000, for example, shrimp farms in southwest Bangladesh. So if you go down, you don't get any money for your, if you go down with disease, you don't get any money for your crop, etc. So that's just the important part of that slide. We've done also lots of workshops and so on. These are actually with finfish farmers in the north who tend to do more kind of single batch stocking and so on. And when we had discussions with them about why they use antibiotics and so on, we get this, well, if the fish all die, we have nothing, okay? A very palpable sense of the shouldering or the burden of risk in that sense. So what does that all mean? Well, the paradox of disease burden, I guess, the burden of being disease-free, is that what we demonstrated was being disease-free or reducing the amount of disease incidence in a farm by changing the seed and the social practices around farming, actually within that socioeconomic setup made acts of desperation more rather than less likely. In other words, best practice management intensified the livelihood risks rather than kind of what's the term, tinkering with your pond, constantly trying to get a livelihood out of it, the idea of moving to a single batch system intensified the risk. So reducing disease incidence in this case would produce the conditions for greater antimicrobial use. So a second example, and I'll come back to these examples and try and get some of the import of them in the final slide. Another project that we're involved in at Exeter with Henry Buller as our lead and with Bristol Vet School and Edinburgh is looking at diagnostic innovation in livestock. And I'll just cut to the chase there. That was, I guess, prompted or was funded because of the O'Neill report, which many of you will know in the UK context was really important and internationally for the designation of how we think about antimicrobial uses. And one key phrase in it that we focused on, for example, is that this idea of empirical diagnosis and the way in which I would say it was extremely pejorative and perhaps derogatory of a set of skilled or other practices. So when professionals decide whether to prescribe an antibiotic, O'Neill said they usually use so-called, and I've just highlighted that because it's an indication of some derogatory comments, they use so-called empirical diagnosis. They will use their expertise, intuition, and professional judgment to guess whether an infection is present, what is likely to be causing it, and thus the most appropriate treatment. In some instances, diagnostic tools are used later to confirm or change that prescription. And of course, that's right, that samples are sent off to the labs and so on, not in a dissimilar way in which some clinical practices are there's a use and then questions are asked later. So why is this important and what are we focused on? Well, we're doing a number of things. We're talking to innovators of pen-side tests and real-time and quick tests and so on, and thinking about what these will do and whether these will in fact change empirical diagnosis to so-called more precise farming, etc. But look at this, just this extract here from a interview with vets, and it's amongst a load of other interviews. In some ways, we're still doing the analysis, so in some ways this is chosen a little bit at random at this stage. But take two examples, and one of them I think is the first one quite close to Paul Osaku from who did one of the first examples. So this quote says basically, we never treat in isolation, it's always in context. A high level of E. coli doesn't mean that they need treatment. So she was talking about UTIs and the fact that if you start just testing for bacteria and then treating, you'll actually over-treat the issue because you're ignoring the clinical signs and so on. The same comes up with the vets. We want to know how do we look at the birds on the farm, the mortality figures, the history and so on. Classic diagnostic behavior then is that you piece together both the bacteriology in this case, but also the history, the observations, the investigations, and so on. And in fact, you may not treat, even though the test says, please do treat, you may not because of that. And then sometimes you may actually have low bacterial counts, subclinical amounts of high levels of sometimes you have to treat subclinical cases because you make a judgment, this is the second part of the quote, that severe lameness can occur later on. So within the contexture of the production facility, it may be actually more efficient to use antibiotics early rather than later because when the bird's heavier, it's less likely to respond, all those kinds of things. So the headline there is that AM uses or choices are not simply the absence or presence of susceptible microbes, but a distributed diagnostic. And I've for years been talking about diagnostic as literally meaning knowing around. Of course, it's the assemblage or the multiple, the practical assemblage of multiple kinds of knowledge bodies and so on into place in order to do to know around an issue or problem. And tests, I think what we're finding from this, and I'm getting close to the end, tests are additional to rather than replacing so-called guesswork, okay? And I'm emphasizing the work of guessing there. It's not really guesswork, it's actually drift work or something like that, some sort of way of assembling lots of different kinds of information and readings together. So what difference does it make? And this is where I'll end and say where, this is where I'm genuinely getting stuck. But I think when we're talking about antimicrobial uses, we need to understand actions, knowledges, and devices as distributed and disseminated within and across sites and situations. Okay, for me, that's my my bread and butter as a social scientist is that's what I would always do. It's not always satisfactory, but that helps me understand the ways in which certain actions get done. So if you change the distribution or the intensity of risk, for example, in a farming situation, despite it being a lower disease situation, you can increase the likelihood that you're going to use antibiotics. So there's no simple kind of match between disease incidence and disease burden. The burden of disease has temporal and other dimensions to it, has a distributional element to it. Interventions then are not just about improving choices, but understanding how actions are pushed and pulled by an array of human and other actors being economic margins, value chains, all those kinds of things. So I think this does two things, 10 seconds. I don't think there are any silver bullets here. We don't have kind of technology to answer the question, but we may avoid promoting the wrong solutions. For example, low disease, but increased livelihood risk food production is one example. I am stopping now. And we can start to identify key levers or intervention points and carefully think about how do we translate the learning across sectors and settings. That's so I told you I'm ending with a little bit of kind of uncertainty as how to do this, but I think that's probably where I'm at. Good, I'll stop.
Fantastic, bang on time, Steve. Terrific, thank you very much. Okay, and we will go straight to the next speaker, who is Klaas.
Perfect, can you see my screen?
Yep, looks good.
That's excellent. Okay, thank you very much for the invitation, Claire. My talk today is titled Pyrrhic Progress, a historical bird's eye view of antibiotics and stewardship in agriculture. So I'll put my top hat on and start going through the talk. So as anybody who's familiar with Claire's work knows, we live in an age of antibiotic infrastructures in medicine and in agriculture all around us. And in many ways, this antibiotic infrastructure or this antibiotic era is already quite old. In fact, many of the key technologies we rely on are increasingly octogenarian. Penicillin and the sulfonamides were developed around about 80 years ago. So this infrastructure is old, but there was this golden period between the 1930s and 1960s when the sheer amount of new antibiotic classes entering the market triggered a real boom of new antibiotic applications, not just in medicine, but also in food production. In fact, the use of antibiotics in food production closely followed their introduction in medicine. By 1940, key veterinary journals were advertising the use of antibiotics to treat disease in individual animals. And in the course of the 1940s, that kind of group of applications expanded rapidly, first to the prophylaxis of disease, so treating animals without any key indicators of disease in bigger groups. And by 1949, there was the claim that feeding low doses of antibiotics over time could promote animals' growth by allowing them to metabolize their feedstuffs more effectively. And this was the so-called antibiotic growth effect, and it triggered a whole new class of non-therapeutic antibiotic use. The results for animal production were significant. Even though antibiotics by no means drove intensification as the sole force, they did facilitate intensification in the 20th century. Farmers on both sides of the Atlantic and actually on both sides of the Iron Curtain used antibiotics to treat animals, prevent disease, ensure themselves against economic losses. So coming back to Steve's talk here, resulting from disease pressure, and tried to boost productivity as a result of increasing these small cost profit margins on farms. It would, however, be wrong to think of antibiotics just as a technology for animals. Antibiotics applications extended across the whole spectrum of food production. They were introduced to plant production from the mid-1950s onwards and played an important role not just in Western agriculture, but also in Asian countries who were intensifying rice production during the time as a treatment against rice blast disease. And finally, for a very brief time, antibiotics seemed to promise a whole new style of food preservation in an age prior to mass refrigeration, when they were used to preserve poultry, fish, and for a time it also seemed that they could even revolutionize whaling by making whale meat more affordable and preserving it. In many ways, antibiotics' rapid spread through a very diverse ecosystem of production, if you think about the wider food chain, can really be explained by their utility as labor. Antibiotics maximize the labor of non-humans by boosting their productivity. They minimized labor losses by enhancing predictability. So by, for example, curbing disease losses and really guaranteeing profits, at least that was how they were sold. And especially by freeing human labor, agricultural and veterinary labor of care during a time of rapid rising demand for rural labor in urban and industrial settings after the Second World War. So it wasn't just antibiotics that acquired infrastructural labor importance, but the labor they were undertaking. It, however, also begs the question, why, if antibiotics were such a crucial labor storm, why contemporaries allowed this labor to spread through so many different parts of the system if there were such a significant risks, such as antimicrobial resistance selection attached to their use? Very simply put, didn't contemporaries know about AMR when they were mass introducing antibiotics to all sectors of food production? The answer is perhaps surprising. Yes, they did know about AMR, as this advert from Feedstuffs from 1966 very clearly shows, but according to the cost-benefit equations of the time, which were very short-termist, the risks attached to antibiotic use were medium to long-term, whereas the gains were very much in the present. So the short-termist logic facilitated the rapid adoption of antibiotics. In fact, public health regulators had been aware of the potential of mass resistance selection resulting from mass antibiotic use since at least the late 1930s and 1940s during wartime settings. And as I show in an upcoming article in this volume to the right-hand side here, CDC regulators were also probing the potential public health risks of mass antibiotic use as early as the early 1950s. However, due to a mix of factors, these early AMR warnings were not taken serious. On the one hand side, commentators remain confident in industry's ability to keep ahead of AMR, and that's something we have to keep in mind when it comes to the likely development of new antibiotics in the future. There was also a lack of epidemiological data connecting distinct environments of antibiotic usage, so connecting the farm, for example, to the hospital. And initially, there was a dominance of vertical, localized scenarios of resistance proliferation, whereby people thought that resistance could only proliferate in a vertical way, in a hereditary fashion. And finally, there was a traditional regulatory focus on the purity of a product reaching consumers, rather than risks resulting from production practices which would perhaps not affect consumers. This risk equation changed dramatically in the late 1950s and early 1960s, partly as a result of the rapidly growing density of British microbial surveillance. You can see behind me here a map of PHLS, Public Health Laboratory Service laboratories densities between 1939 and 1965. And the data generated here was very much also integrated into knowledge produced by the Central Veterinary Laboratory in Weybridge, and resulted in instances where regulators could link distinct instances of antibiotic usage with risk down the line for humans. The risk scenario increased extra proportionally in 1965 as a result of a paper by Ephraim Saul Anderson and Naomi Datta, which described R-factor transfer in non-human reservoirs. So resistance could not only spread vertically in a horizontal fashion, but could actually be exchanged between different bacteria via the exchange of small plasmids. Did this actually change the wider ecosystem of regulation? No. One of the reasons for this was that AMR scenarios were much too abstract to galvanize public and political action that was pronounced enough to break this existing logic of short-termism, whereby, again, regulators, but also industry actors repeatedly managed to push through a view whereby short-term economic gains were given priority over very vocal warnings by some actors of medium to long-term risks. Even where warnings triggered reforms, resulting reforms remained very narrow. A classic case example for this is the British Swan Report. Published in 1969, it's often seen as the first call for precautionary antibiotic restrictions, but if you actually look at it more closely, it's a very narrow attempt to fix antibiotic infrastructures rather than to reform them. The Swan Report called for a reservation of therapeutic antibiotics for prescription-only use, but in effect only really changed at the very margins requirements for dosage and classification for many antibiotics without really reducing access. The subtherapeutic penicillin and tetracycline feeds were banned in 1971, and Britain and later Europe aggressively promoted the Swan Gospel abroad, but there was no underlying reform of infrastructures. Agricultural subsidy and intensification policies went unquestioned. Ersatz antibiotics like virginiamycin were provided, but were not really tested for their effects on resistance selection, and veterinary income systems were not targeted for reforms, thereby failing to curb profit motives for the prescription of antibiotics. All in all, the Swan reform was a classic case of a narrow reform that failed to reduce both antimicrobial usage and antimicrobial resistance. And it's not unique in the history of antibiotics either. In fact, the history of stewardship is littered with narrow reforms that fix specific parts of the infrastructure, often serve to boost the interests of specific interests within the wider ecosystem of the antibiotic market. In the Swan report, this were VETs and ersatz therapies, and once established, proved very difficult to dislodge because these vested interests defended this narrow reform trajectory. They also had very limited appeal outside of the high-income settings in which they were pioneered. In fact, from the 1970s and 1980s onwards, what we see is an increasing fragmentation of the global ecosystem of antibiotic regulation. In the US and many other American countries, as well as east of the Iron Curtain, there was an ongoing focus on intensifying agriculture and producing new forms of intensification, such as feedlots for cattle, aquaculture in the Scandinavian, but also North American and South Asian context, and in East Germany, also a focus on intensifying pig production. At the same time, antibiotic use was rapidly expanding as a result of rising meat production in the so-called global south. And behind me, you can see a graph from China and Brazil indicating a rise of meat production and antibiotic use and giving rise to a whole new raft of massive companies which integrate antibiotic production and meat production. This was in part driven by genuine reforms within these countries trying to modernize agriculture, and also as a result of aggressive pushes by Western companies into these markets, with the result that centers of policing, as Christoph Grabmann have called them for antibiotic stewardship, no longer aligned with centers of production from the 70s onwards. The result of fragmentation was not only a failure of international stewardship efforts, but also really highlighted the fallacy of thinking that prioritizing AMR risks is inevitable across cultural contexts. Stewardship appeals were consistently meaningless outside of HICs and even within HICs if they were not made meaningful to multiple stakeholder groups. We can think this through in the case of the most recent wave of stewardship reforms, which started from the mid-1990s onwards, first in Europe and then in North America, and were based on both statutory and voluntary interventions into the antibiotic market. Resulting reforms have, in both the UK and the US over recent years, led to impressive reductions in non-human use relative to the high points of usage in the 1990s, but are still at a higher volume of consumption than during the first era of antibiotic use when problems became apparent. Their impact on AMR also remains unclear, and most importantly, it is completely unclear whether these solutions will work outside of high-income contexts, something that work by Steve, for example, is highlighting. So what can history do in this context? First off, as I hope to have shown, the history of AMR and control measures is older and more complex than is usually portrayed in the policy reports, which usually focus on the last 10 to 20 years. What we can see historically is a succession of short-termism, narrow reforms, and epistemic fragmentation produced by regulations that attempted to fix individual aspects of our antibiotic infrastructure rather than addressing it holistically. Historians in this situation can take off their top hats and help by understanding the longer-term drivers of antibiotic use and the limits of our understanding of AMR, not just with regards to diagnostics, but also in terms of cultural understandings of AMR, and inform policies that are both structural, equitable, and tracked. And if you want to get an idea of what that means, you can check out a recent paper which many of the people on this panel were part of. Where next? And I'm coming to the end here. Well, I think we've now got an increasingly well-studied body of literature on higher income countries. What we don't really understand is how antibiotics entered middle and low income contexts, and how in this context they develop very distinct path dependencies of usage, understanding, and knowledge and regulation. And we really need to understand this, not just for individual case studies, but in regional contexts to understand how knowledge, microbes, and drugs flowed. We also need to understand the very complex history of market-led and statutory stewardship. And this is becoming increasingly important as a result of assurance schemes, which are becoming international with the help of labeling and organic reforms and production systems that are basically outsourced from high-income to low and medium-income countries. And finally, we need to analyze the emergence and evolution of antibiotic stewardship as a topic of international governance as a historical object, and really ask whom stewardship serves at the international level. And if you want to learn more, here are 2 books I highly recommend. Pyrrhic Progress was published this year by Rutgers. And if you want to learn more about assurance schemes, Bearing Witness on the History of British Animal Welfare will appear with Palgrave in March next year. Thank you very much.
Excellent, class. Thank you very much. You sped up to just about, so I didn't have to cut you off. Fantastic. I'm going to go straight over to Salah, if you're able to share your screen.
Yes. Now. Okay. Thanks very much. Thank you, Steve and class, for the great lead up to the talk. My presentation follows very nicely from the kind of the call for attention, what happens in the low and middle income countries. And I'm presenting a shared talk developed by Andrea, and together with Andrea Butcher and Jose Cañada, with great acknowledgements to a huge range of collaborators in West Africa within social sciences and microbiology that I would like to acknowledge. We're going to tell you about our ongoing work in West Africa regarding the spread of AMR genes, and that this is work that we're doing as part of a larger consortium together with clinical and environmental microbiologists, where we're sort of contributing by looking at the situation on policy, animal environmental levels regarding the social practices that might drive antimicrobial resistance spread. And this is very much work in progress. So you're getting a sort of glimpse into our preliminary findings or some preliminary thoughts at this stage of the data that we're analyzing at the moment. So this work is based on quite an extensive range of field work, where we've tried to look at a very broad set of different kinds of practices, context, discourses with regards to what might be contributing to the spread of AMR genes. That has included focus groups, participant observation, interviews, documentary analysis, and questionnaires, and the work is very much ongoing. But the field work has been taking place in Burkina Faso and Benin, which according to the Global Health Security Index are among the least prepared countries in the world. Both countries lack a robust surveillance and evaluation mechanism, and they don't report to the GLAS about antimicrobial resistance. Yes, so Africa is presented as a place that from where there is a lot of gaps in data with regards to AMR, and the 2 countries have been setting up their national program action plan for antimicrobial resistance, but which hasn't been sort of nationally validated as of yet. So work is ongoing to set up different kinds of structures to monitor and control and prevent the spread of AMR, but we're not they're not quite there yet with this these action plans. Antibiotics are widely used in place of healthcare infrastructures or veterinary infrastructures. We'll tell you more about that. But antibiotics are both used extensively for human and animal consumption, and can be purchased over the counter and are regularly bought in informal markets through chains or flows of drug trafficking from the Nigerian side. The healthcare, different kinds of infrastructures with regards to sanitation and hygiene are very inadequate, let's put it that way, and that you'll see some of that evidence in the presentation. So what we're going to describe you today, it comes from a much bigger range of different kinds of observations, analyses, themes, patterns that we've identified. But with the purposes of time, we're focusing on 2 specific thematics, 3 specific thematics in a bigger kind of pattern that we're called stacking of lacks, or an array of lacks, whereby when you think of what happens in a particular context, it's always followed up by yes, surveillance doesn't work. There isn't any data because there is no surveillance, because there is no diagnostic. So it amounts to this kind of layering of different kinds of lacks that I will tell you a little bit more about. But so that this is a sort of the pattern that we're highlighting with these examples. When thinking of ecologies, what the challenge that we're faced with is that the AMR genes have this ability to travel across the different kinds of situations and contexts. And a big factor in the way in which that's relevant to this particular discussion today is how antibiotics are used within animal husbandry. And the examples we've selected come from poultry farming, whereby it very much sort of chimes with what Steve and Klaas described as using antibiotics to increase margins. But in the cases where the fieldwork has been done in these poultry farms, antibiotics were not necessarily used to increase margins, but rather ensure that there was any money to be made at all. For example, in this particular farm that had 20,000 hens producing 12,000 eggs per day, if the farmers identified, or the carers, farm owners, if they identified a disease, then the whole farm was treated. And in abiding by withdrawal periods, they should have then not sold those eggs for a period of time, which would have resulted a huge amount of loss to the farmer, plus the knock-on effects on the customers. So as a result of that, these guidelines of withdrawals were rarely followed, because it would have resulted in major risk to losing their entire flock or the income that could be made from those birds or the eggs. So in that sense, it wasn't about increasing margins, it was about having any livelihood at all. Similarly, antibiotics in this poultry farm, it operated as a way of ensuring that the birds could live. So this farm was not as well maintained as the one in the previous slide, and the owners or the managers were not as trained as in the previous case. What then happened was, when birds started to look like they were not doing so well, they were given antibiotics up to the point that the antibiotics ran out. And when the antibiotics ran out, the birds ended up dying. Here, the antibiotics were then the assurance or the possibility to make any kind of income from there. And it served really as the infrastructure for being able to continue to farm this particular breed of chickens to begin with. Both of these cases, what they highlight is the lack of access to diagnostics, and where the treatment with antibiotics stood in place of any kind of testing. The quote here is from the human side, clinical diagnostics, but there was a pattern which was very similar between the veterinary health and human health, whereby doctors or vets simply said that there's no diagnostic tests available, so they were using what Steve referred as the kind of the hunches, based on which they would have then rolled out antibiotics. Often vets were not actually invited at all, but this guesswork from the report, O'Neill report, was done by the carers of the farms, whereby they, based on their experience, were gauging if something was wrong, then antibiotics were rolled out. So there was a lot of professional and quote unquote unprofessional, in the sense that non-trained medical experts, veterinary experts, were taking decisions on what was going on in the flock. Because antibiotic use is a small factor that creates antibiotic resistance, antimicrobial resistance evolution, an important factor or vector in this is the environment where microbes lateral gene transfer takes place and microbes spread genes. So the antimicrobial resistant genes can go from one bacteria or virus to another without necessarily there being the antibiotic use itself. And we've done some work with the clinical environmental microbiologists to look at sort of putting together the social practices and the
1 minute, Sarah.
Okay, perfect. The samples, and water plays a key role there in the sense that water both acts as a hotspot where this lateral gene transfer can take place, but it's also a medium that can travel across lots of different places. And people have very elaborate methods of gauging whether water is safe or not, but in the absence of safe water or what is perceived as safe water, they then sort of resort to using water that is perceived less safe, because of water is needed, or not use water at all. And what this comes back to, and this is my 1 minute last comment, that for the solutions, that what the fragmentation that Claes so well described has resulted in a difficulty of saying, well, where do we start in a situation like this? There are livelihoods at stake, there is no diagnostics, water treatment. The case of the example from our microbiological findings showed that there are a lot of resistant genes for, especially in hospital water, which is supposedly treated, but yet a lot of AMR genes are present. And they are looking at this list that is just from these two examples, the list of different kinds of things that would need to change, if you will, or that contribute to AMR resistant gene spread. Is just so overwhelming that this amounts to the stacking of lacks that we explained, and there's no silver bullet, but that very systemic complex changes would be needed to address this. Thank you.
Fantastic. Thank you very much, Sarah. Gosh, we've got so much to think about and discuss on panel. It's going to be great. So we're going to go, so I'm going to share the background one for this one. Hang on a second. How's it gone? Can we go to original? Oh, how am I? Apologies. This one has disappeared. Sorry, my PowerPoint has frozen. Sorry. It's not a good time for that to happen. Okay. Right, it should now. There we go. Okay. Over to you, Ridgel.
Great. Does that mean I get an extra minute? Or
You definitely get an extra minute, bonus minute from my mucking around.
thank you. Thank you, Claire, for this invitation to speak to you today about my ongoing doctoral work. I am the youngest and the junior most amongst the panelists today. I'm still pursuing my PhD at MIT, where I look at not only the sort of broad cultural issues raised by the problem of antimicrobial resistance, but also what people are doing in terms of epistemological and scientific labor in the face of this problem. I'm an anthropologist and historian who has conducted fieldwork on 3 continents, in India, Georgia, and the United States. And I've done so with the intention of trying to reveal the deeper histories that are implicated in scientific enterprise, in particular the sort of cross-continental and cross-cultural histories that are implicated in the story of bacteriophages. In the last 4 or 5 years, social science research on antimicrobial resistance has done a great job in laying out the truly multifaceted nature of this problem. AMR, as we know now through this research, is simultaneously a cultural, ecological, and an ethical problem. And so a vibrant community of researchers housed at the AMIS group and the Antibiosis collection at Nature, but also scholars who are dispersed across Australia, the US, and Europe, as well as India, and Kenya are trying to grapple with the immediacy of this problem, both in a local context, but also on a global scale. Antimicrobial resistance, like climate change, is one of those problems which is planetary in scale, but also one that is experienced in locally singular ways, and in ways that are asymmetrical. Some places bear the brunt of this problem in lesser fashion than other countries that are more resource poor. What this research has done has shown us that that cultural variation precludes us from approaching antimicrobial resistance as a problem that deserves a singular response. It is, after all, not a problem of novel bugs, novel drugs, and rapid diagnostics. In fact, the social studies of antimicrobial resistance, to me, suggests the revelation of certain double binds, where on the one hand, you need immediate cure and relief for symptoms and diseases that people are living with, versus on the other hand, one has the mandate of what I call antimicrobial responsibility. And we find through this social science research that the need to respond to emerging biological contingencies is sometimes much greater and much heavier than the mandate to antimicrobial responsibility. I have some personal experience of this. My father is a poultry farmer in the east of India in a place called Assam, which is famous for tea. And having lived and been brought up on a poultry farm that has often been invaded by all kinds of biological and viral pathogens, one understands the sort of the heavy burden that people like my father have operated under. And so antibiotics in that kind of economic and epidemiological setting offers not an easy fix, but one of the few tools or weapons that people with limited resources have at their disposal. I also have friends with disabilities that I have lost to antimicrobial resistant infections in hospital settings and at homes, and I understand quite viscerally what it means to use and to deploy antibiotics much more rapidly than what the discourse of antimicrobial responsibility would have us believe. Patients who are desperate for cures in resource-poor settings, for instance, are often drawn to doctors who prescribe the latest antibiotics, because antibiotics do have a certain cultural capital within them. And so not only are these tools or weapons that are important for people like my poultry farmer father, or for friends with disabilities who have struggled with and perished at the hands of antimicrobial resistant infections, but also for people and for doctors who regard these as magic bullets even today. And so I have been driven to investigate what people are supposed to do in the face of drug-resistant infections. What is the everyday life of antimicrobial resistance like? I ask in my research. And to answer this, I was first drawn to it as a diagnostic problem in human and animal or veterinarian worlds. I wanted to explore ethnographically the real paradox that our increasing diagnostic prowess to diagnose drug-resistant bacterial infections comes at the very moment where our capacity to respond to those pathogens is diminishing. And so this begs the question that a lot of us are asking today, which is that, is the antibiotic era merely a blip, an exception that proves the rule in the longer history of the relationship between humans, animals, and pathogens? So I conducted some fieldwork in a microbiology laboratory in Delhi, where I wanted to understand the scientific and technological nuts and bolts of what goes into culturing pathogens on a petri dish and then figuring out their susceptibility or resistance to specific antibiotics. I wanted to understand the ground level contingencies under which scientists and technicians in this lab operate, and what they make of the fact that their labor is increasingly being de-skilled at the hands of pathogens that are resistant to cures. They can increasingly diagnose pathogens, but they can't, they are at a loss when it comes to prescribing drugs to battle those pathogens. And so through that laboratory where I met doctors as well as patients who could diagnose but no longer treat infections, I was connected to the Eliava Institute in Georgia, where people were proceeding along the lines of what I like to think of as the pursuit of technoscientific salvation. When patients become pilgrims, as I write in my dissertation. People were desperate for cures in settings like India, some of them friends of mine who have either been treated at the Eliava or who died before they could be treated for their drug-resistant infections. And I ventured there in 2018 to try and figure out how Eliava's scientists today are emerging from the devastations of the Soviet Union's collapse to promote bacteriophages as technoscientific salvation in post-antibiotic worlds. It's also a very interesting moment where, or rather, it's also a very interesting space where scientists who began as medical or medical interns or scientific trainees at this institute have come of age and become respected scientists in their own right today. And their life story spans the life story of the emerging post-Soviet Georgian Republic. And so in some ways, one can tell the story of the last decades of the Soviet Union and the first few decades of the post-Soviet era through the life stories of these scientists. But fundamentally, I'm most interested in the ethnographic question of how knowledge and history travels across time and space. And this is the question of the epistemic trace as it unfolds across place and species. And so this article that I wrote in the Notes and Records, which I was glad to share space with class, raises the question of not only Georgia as a place of technoscientific salvation, but also the question of the epistemic trace. Where does the bacteriophage emerge from? And if one were to delve into its interesting history, often one finds that one begins with Felix Derel and his work. And there is often a footnote that traces it to Allahabad, where it says that Derel's work in the early 20th century with the discovery of bacteriophages was influenced by his reading of a British bacteriologist by the name of Ernst Hanbury Henkin, who did work on the sort of so-called magical or curative properties of the Ganges waters in India. And I wanted to explore not only this historically, but also ethnographically. Is it more than merely a footnote? Is there more to the story of bacteriophages in Allahabad, which is where the Ganges and the Yamuna rivers meet, which is also the place of the Kumbh Mela pilgrimage, regarded as the largest religious pilgrimage in the world. And I wanted to explore what bacteriophages and what I call its pre-discovery in this space means for practitioners and for lay people today. How do various practitioners such as biologists, local phage biologists, historians, Hindu priests, Ayurvedic doctors, biomedical specialists, how do all of these people surrounding the figure of the virus in the rivers incorporate it within their everyday life and culture? And that's the question that I've sought to answer in this article. And ultimately, one comes to the conclusion that like antimicrobial resistance is a cultural problem, the history of bacteriophages is also a cultural one, one where secular and sacred views of illness, disease, and healing, where scientific history, where Ayurvedic and biomedical modes of treating infections and religious myth come together in very interesting and novel ways. And so all I will leave you with is by suggesting that bacteriophages in the context of AMR is still an emerging sort of tool, but it is not just an emerging tool. It is also a tool that incorporates within it a whole host of conflicting histories. And to tell the story of bacteriophages today is somehow to tell the story of what it means to be human in the early 21st century. Thank you.
Fantastic. Thank you so much. I'm going to stop my share. So what we will do now is we're going to go to a short hour panel discussion. Actually, I'm going to try and stop the share so that we can see each other. Fantastic. Wow. Huge number of questions arising that cut across, you know, themes cutting across your talks. And I mean, I guess what we're hearing is, you know, if we pose that question of, so what do we learn from social science, a range of different types of social science we've been talking about, you know, the anthropological perspectives, historical perspectives, different places around the world. And I think, you know, if we're going to answer that question, what do we do about antibiotic use? We're hearing from, you know, the last talk with Rajul, you know, thinking about, well, you know, and that appeal, and I like your phrasing of the techno-scientific salvation, you know, this bind that we find ourselves in, could the answer be in having bacteriophage? Would that now solve the problem? Can we use that instead of antibiotics? And that idea that well, that would get us out of this struggle that Steve started us off with, which is actually, this is really a really hard thing to address, because you know, in Steve's talk, when he's talking about this challenge where we're stuck between, you know, an individual choice, we make it an individual choice problem, or we tend to start talking about those the structures and the lack of choice. And I think, you know, that was mirrored in some of the things that was said, you know, through each of our talks. We were hearing this kind of challenge between you know, individuals, structures. You know, your point about the magic, you know, the potential magic bullet, one magic bullet, you know, replacing another, perhaps. You know, if you start to think about bacteriophages as that solution. But I also wondered whether there's something there with, because of that different history that you were just talking about, Rajul, in terms of bacteriophages, they, you know, because they are scripted so differently, and we'll hear in our next panel, Charlotte Breves is going to be giving us a talk, and she's been tracing out some of this, you know, this resistance to the idea of bacteriophages being seen as antibiotics, because of some of this history that you're talking about. Does it then come with a different kind of mid-level scripting that could actually stand to give us some challenge and render more visible the things that are meaning that antibiotics have become so inevitable, that's in this middle level that Steve's kind of suggesting that we start to look at, and I think was mirrored in Salah's talk and classes as well, that, and all of the talks about that kind of, you know, trying to say, you know, is there something in between a individual choice and structural model where we're, you know, looking at those networks, where we're looking at the materials and the fabric of make us do what we do, but not only the material dimensions, but the scripts and the guidelines, and I thought perhaps the regulations, as Claes is talking about, those all kind of script what happens, so that that's sort of not, it's neither an individual choice nor necessarily a political economic structure, but it's somewhere in between. And I guess, I mean, you know, Steve started off by saying, I don't know, you know, it's easy to get stuck here, and what's the answer? But having, you know, I feel like that in many ways there are, you know, all of us are asking that, saying, is there something more to be learned from that space about how we can think forward? And one provocative question I'm going to throw out, emergent from that, and I think it's provocative to us as a group of scientists, social scientists, and forgive me to those on the call who are more behavioural economists, but, you know, and I'm slightly hesitant to ask this, but is there a role for nudge in that middle space? There are nudges that are very nudgy, you know, behavioral scientists, nudgy, very close to behavior, but then there are these nudges which are about making your tax forms easier to complete, And there are these kind of things that are in the environment in between that are about changing the systems. To what degree, and I'm not necessarily saying it's the right answer, but to what degree does what we end up talking about start to overlap with that? And what do we need to be careful about there?
I think the first part of your question, if I'm reading correctly about the scripts, reminds me of the work Hidden Transcripts by James Scott, which argues that certain actions are neither to be located in the individual nor to be located in the system, but in the relationship between the individual struggling against the system and coming up with various strategies. And I find that the story of bacteriophages today, especially as it unfolds when people take these journeys to travel to Georgia to receive treatment for various sorts of infections, is really one of those, the journey itself is one of those sort of modalities by which the individual struggles against the system being one where bacteriophages are not recognized. Bacteriophages are not allowed to be transported easily. There are no antibiotics that people have access to that might be effective for certain kinds of infections that they might have. And so these are those ethical plateaus, right? A point in time when people don't necessarily have too many things at their disposal, which is why they undertake all kinds of hazardous journeys and expensive journeys. What I am seeing at the ground level, though, are increasing is what happens when people come back and they become converted to a certain cause. I'm not trying to promote phage therapy, but I do find that people, when I speak to them, especially after they've returned with some relief, symptomatic relief from Georgia, often are in the pursuit of trying to expand access to what they themselves have had access to with great difficulty. And so one finds increasingly interesting collaborations between labs in the institute in Georgia and labs in places like Delhi or Kenya or certain countries in Western Europe, where they're coming up with interesting ways of diagnosing infections locally, but prescribing bacteriophages cross-continentally. And all being done in the pursuit of trying to reduce the costs and the time that is involved in this therapy.
And also in the pursuit, there's so many other things that are gained. There are so many other political things that are gained from that pursuit, which is reminiscent of what Klaas has described from the antibiotic side as well, and the other things that get carried with it. Would Steve or Klaas or Sado, any reflections on the question of how we, if we drill down in this middle level, where does it take us? Klaas?
Perhaps that there's no return to a kind of age of innocence. I think some of the tensions raised in these talks are absolutely spot on, that there is no either or. There's no either focusing solely on the individual or focusing on the structures, but rather since 1945, antibiotics, phage, also as tools of biotech now, it's not just the Liava, right? They're also heavily commercialized. You will find them on the US West Coast with lots of patents on them too. That all of these are connected to a world that has become much more interconnected, and where these regional flows of disease and of therapies are heavily interlinked. And there's connected to this your question about whether it's foolish to ignore nudge theory here in this regard. Yeah, of course it's foolish to ignore nudge theory when it comes to specific instances of changing behavior. But I think what is much more productive, rather than always seeing it's either structure or changing individual behavior, is to think of the fact that we will not return to a world without resistance. We will not return to a world where the antibiotics will suddenly become all effective again, or where we will have a bright new age of endless supplies of new antimicrobials, but rather that we now live in a world where we're dealing with supply chains which no longer follow these classic nation state narratives that in the social sciences we still love to tell ourselves, and that we live in a world of market pressures where consumer choice can have a significant pressure on markets in other parts of the world. And reflecting on that and finding new archives and sources to study, for me, is the way forward. And this is one of the three challenges I raised, was to focus on voluntary versus statutory reform, because that seems to, for me, to be the big impasse also at the international level, where we have international systems that focus on modifying nation state behavior, whereas anybody who studies the market flows, anybody who studies agriculture knows that in a way they don't actually have as much power as some of the larger companies that I showed at the end, some of the larger Chinese companies, for example. So finding new ways to address those flows and leaving behind these contexts for me would be a big answer to reconciling perhaps nudge with structural solutions.
Yeah, yeah. And in some ways what you're saying, I think, sounds a bit like we're saying we shouldn't be putting human behavior at the center of the model. That by putting, making antibiotic use be about human behavior, we end up asking certain types of questions that circulate around that. But you're saying perhaps we need to be, putting something, decentering behavior in there. Salah, were you going to comment? Oh, Steve is unmuted. Go ahead, Steve. Sorry.
No, I just want to agree with that. I think that's absolutely important. I don't think these are different levels. I don't think this is structure at the top agency and we need to be somewhere in the middle. Otherwise, I start sounding like a kind of throwback to Tony Blair or something. There's a middle way here, which would be dreadful. But the middle is etymologically associated with the muddle, and that's why there's a complex present to these issues, and it needs those kinds of approaches. I think actually behavioral economics, and that's why I started with Dusenbury, could be a sort of father of, or one of the parents of that approach. There is something there. But I think as soon as you get beyond the kind of relatively simple issues like, a public health message around smoking or sugar, that kind of thing, I think you start to get into some real difficulties and it doesn't kind of work as well because you do end up reducing human experience to behavior rather than all the other things that we've talked about. I think Klaus is absolutely right, as well as that these corporations are absolutely key. And if we look at the experience the veterinary medicine director have just produced their results for last year. It's clearly, the key actors are people like the British Poultry Council, Arla, other companies and so on that are acting. I think what we've noticed though and maybe Salah can come in on this, that kind of approach is difficult when you're in somewhere like Bangladesh or maybe Burkina Faso, partly because global trade is changing quite quickly as well. So we have increasing South-South relations now, where some of those levers that were talked about, the ethical consumer or the virtual consumer imagined by someone like Arla, because they never really asked anyone, but they work with an idea of virtual consumer. It doesn't work as well. We're just doing a project in the moment in Bangladesh on pharmaceuticals in agriculture and so on. We've got no evidence at all of anyone in even Dakar or the growing middle classes having much of an interest in AMR as a kind of issue. And likewise, as the international trade patterns change for Bangladesh, then you start to get different kinds of relationships, which may unsettle that. China's the big question, of course, but I mean, there's a whole set of issues there, I think. So again, going back to Rigel's point, that this is, I love the line, it's planetary in scale, but experienced in locally singular ways. And I think we just need to keep that on the agenda because it's difficult to find one solution that fits all these different contexts.
Thanks. Salah.
Yeah, I would like to follow up on the point, Claire, that you ended up your point with, which was about sort of questioning the human behavior element in this, and to follow on from what Klaus and Steve said about like not thinking of the agency as either at the level of the individual or then pushed down by some of the structure. I think what do we mean by structure there really? Is it a question of someone having to give antibiotics to their animals because they don't have livelihood options, or are we talking about the sort of systemic approach in healthcare where people are just handed out antibiotics? I think it would be so that what structure can mean in that situation is really diverse. But I would suggest not to look at actually on that level at all, or keep that. What I'm going to try and say is that to understand antibiotic use and antimicrobial resistance spread in particular, we should not focus on the factors that drive people to use antibiotics as the determining factor. And I'm taking sort of Collignon's and others' article about the correlation between antimicrobial resistance, antibiotic use, and what they call anthropological factors, which is much broader than what the anthropologist, social anthropologist would think, but it is very much a quantitative analysis that includes level of education, healthcare, sanitation infrastructures, it includes corruption, level of education, lots of different kinds of sort of quantitative metrics. And they say their conclusion is that it's not the antibiotic use level that correlates with AMR, but it's actually the kind of level of education, investment in healthcare, and so on. So it is these very much sanitation and so on. It's very much these drivers that we could call or lump as developmental, that are the ones that predict the scale of or the level of antimicrobial resistance. So it's because of the lateral gene transfer that bacteria are able to do, transmitting the genes from one another, the antibiotic use only puts a certain amount of selection pressure for the resistance to mutate, to come about. But actually the fact that these microbes can travel within, for example, water, and then through the lateral gene transfer actually spread out, and then what we need to also pay attention to is precisely those factors that the Collignon and others pointed out. So like level of education, the corruption, healthcare investment, and so on. So in summary, what I'm saying is that let's not just look at antibiotic use when we think of antimicrobial resistance spread, but pay attention to factors that are not about antibiotic use as such, but very much in that category of development much more broadly. So the interventions that we should think about have a lot to do with not only how many antibiotics are given to humans or animals, but that whether the hot spots or situations exist where the resistant genes can spread. So I'm sure if we, as social anthropologists, ethnographers, would look at the factors that come under that development would be perhaps more fine-grained than the ones listed in Collignon, and more situated and localized and so on. And yet I think the argument still holds that we need to look at things that go over and beyond the individual user of antibiotics to these very diverse dynamics that have seemingly nothing to do with antibiotics, but something to do with development more broadly. That is where the dynamics is not limited to antibiotic use, but contribute to a lot of other things as well.
Yeah. Steve?
I think that's really important, and it's an important paper, and I've seen people present it that in higher income countries, use is the kind of dominant issue. In lower and middle income settings, often at certain thresholds, and they can get overly kind of prescriptive about this, but transmission becomes the key issue. The problem is, and maybe Claire, because we were talking about this last week, the problem is the same behavioral approaches get taken to WASH programs, to reducing transmission. So for example, work in Tanzania at the moment on the Maasai are trying to get people to not drink unpasteurized or raw milk, because that's the key transmission route. So you realize in a climate changing setting, etc., this is work at the University of Glasgow, which is excellent, and the University of Washington. In a climate changing setting, it's wrong to tell the cattle people to not give antibiotics to their cattle, because they're getting more and more stress, disease, and so on. There's no veterinary infrastructure, etc. One thing to do then is actually then address the way in which those antibiotics then are transmitted to people through that consumption route, and then ends up with neonatal babies, etc., suffering from resistance. So that's one of the key things. But you end up in a similar sort of social science bind in some ways, and I guess that would be my worry. But you're absolutely right, Sala. Transmission, I think, becomes the key issue in lots of the settings that we're talking about.
Yeah. Ritual.
I think this also raises the question of biocapitalist value, in the sense that the question of nudge is not one that only incorporates individuals who are patients or sufferers as opposed to structures, but also very real pharmaceutical companies and biotechnological firms in places like Boston that refuse to invest in new antimicrobials unless certain kinds of governmental nudges are present. These push and pull mechanisms It's interesting that someone in the audience asked a question about nudges, because the word nudge is precisely the word that tech bros here deploy whenever one asks them about the paucity of new antimicrobials. And their point is precisely that there's no profit to be made in these drugs. And so the question of nudges is also one that must be deployed in nudging inquiry towards corporate value and asking, especially in this time of a pandemic, that what kind of corporate or economic system exists where investment in infectious diseases, in vaccines, in new antibiotics is regarded as unprofitable. What does that statement mean? And what sorts of values are implicated in that kind of absence or refusal to invest? And also who invests, who profits, and what kind of incentives are needed.
Mm-hmm.
But Klaus is absolutely right. We're not going back to there's no going back to the future where there is going to be no resistance. But one, as social scientists, one has to constantly ask these questions about what sorts of social norms and values prevail, even in which things like antibiotics circulate.
Yeah, it's such an important point. I'm going to ask if we can add a couple of panelists to the panel, who are the other co-authors from Salah's work and incidentally Steve's. Would you mind adding Andrea and I think Jose? Hi, Andrea. Hi, Jose. Thank you for joining the panel as well. I'm just going to go to, we've got about 10 minutes left. So I'm just going to go to, there's a couple of questions we've seen in the chat. So Nicholas Fatane has written a question which he's addressed to Stephen Klaus, but we can discuss more generally, which is where he's saying, do you think the narrative, considering that overuse is related to intensive and industrial agriculture, leads to the assumption that the current reduction of antibiotic use is associated with transitions towards more sustainable forms of agriculture, whereas it's actually not? And if so, in which ways can research feed this critique of current AMR policies and stewardship programs that generally produce changes that actually reinforce the intensive and industrial forms of agriculture? So I think Steve's shrimp example is one that emphasizes that. Would it, Klaus, you unmuted? Would you like to comment?
Yeah, I mean, I think the question is excellent, right? I think a large part of the reductions of AMU in high-income settings have been driven by an increase of biosecurity on lots of the farms that we've seen, where better prescription practices, better access to diagnostics, but also really improvements in isolating animals further from their environment. And there's an excellent book, Porkopolis, for example, that really nicely describes some of these trends, specifically in the US. I think rather than just saying, oh yeah, so reductions always drive intensification, I think it's very interesting to ask who the industry is who's providing us the counter narrative of purity and better production. So the organic industry, which in a way is a product of the antibiotic age, in as much as it seems to offer a kind of counterfactual alternative reality, right? Organic produces this concept that's been very successful in the West of purity, antibiotic-free, which is strongly culturally associated, which is, I guess, why it doesn't have such an appeal, for example, in middle and low-income countries to the same extent. These industries also have a kind of stake in I think prescribing a popular image of what reform should look like that is often quite distinct from what reform in the antimicrobial area actually looks like, if you look at how farmers have managed to reduce usage. So I think that is actually the real question here. Whose image, bucolic image of a countryside and rural pasture are we buying into when we're structuring the reforms? And the organic industry is a very interesting case sample there, because on the one hand side, they do drive reforms, they do drive new forms of best practice in the 70s and 80s. But from the 1990s and 2000s onwards, they also become integrated into the exactly same supply chains, the exactly same ownership structures, which in a way have perfectly split up the market. The owner owns both the polluter and both the promise of purity, right? And sells both at the same time to the same consumers. If you look at the big ownership structures that affect assurance schemes around the world. And that for me would be, again, a fascinating topic for historical and social sciences research, to really look at both sides of the industry at the same time. So not just to look at intensification versus allegedly non-intensive organic production, but really at the same time study how agriculture after the 1970s, 1980s has evolved to deal with market segmentation, supermarkets as new actors who have come in. I would say, for example, the history of Tesco would be fascinating in this regard. As a supplier both in China and the UK. So for me, that would be a really promising way to tackle this issue that Nicolas has raised. And I think that's often quite disconcerting to the general public if you say, well, actually, it does lead to more intensification in many settings.
And I like the way that this, you know, attention to something like intensification is a process. So you're not kind of saying this is about behavior of somebody here or there. And it's not necessarily saying something so meta that you can't do anything about it. But I think there's been a lack of encouragement for social scientists. There's been a lack of framework for social scientists to interrogate things like regulation. I mean, certain social scientists do it very well, and you know, international relations and so on. There are social scientists who work a lot on some of these things, but then the anthropologists are brought into, you know, look at local conceptions and belief, and it means that we end up with this very skewed sort of database of research, which is, you know, focused on what people believe in. And it sounds from our panels that it's, you know, there's a lot more that's going on that we just don't spend enough time paying attention to about, you know, the regulatory frameworks, you know, what industrialization of farming actually is. I'm just going to see whether Andrea or, sorry, Klaus, yeah.
Just 2 points on that. Basically, what I focused the last 5 years on regulation on the nation state, but I realized that what I was missing out in some of the story was really cui bono, who was winning and who was owning. And those are two very simple questions. They don't require a lot of theoretical sophistication when you're answering them, but I think those are essential questions when you start dealing with processes that go beyond the nation state and really are trying to capture a kind of global process.
I just want to invite Andrea and Jose if you had anything else to add to the conversation that we've got going on. Andrea.
Jose may also just want to say a little bit more, but thinking about regulation, I mean, the situation in the West African countries that we're working with is that they are part of the West African Economic Trading Bloc, UEMA, or the union. And there are regulations about where you get your medicines, your veterinary medicines, you know, using antibiotics and all of these kinds of things. But one of the problems we're finding there, you know, and you've got your your various ministries who have their various extension work and, you know, responsible for feeding these regulations out and training people in how to use them. But of course, the problem is very few people are connected. They're not very well linked into these ministries. There are these registration processes that small breeders will either not know about because they haven't had that communication, or they will avoid because they've had a bad experience, either through corrupt officials, or, you know, these hidden costs that end up coming in, or, you know, so they haven't been able to access the loan system the way that, you know, all kinds of things. Well, not all kinds of things, but those are the various narratives. But so very few of them have any idea that there are regulations that exist. Some people understand that there's a problem with antimicrobial resistance because they've come at it from different ways. But what there really is very, very strong is this, they are so well networked with each other. There are some that do like to stay away, that there's a, you know, certain trust issues. But in terms of people looking things up on the internet and discussing with each other and sharing, you know, forming WhatsApp groups, sharing photographs and that kinds of diagnosis. So, you know, the diagnostics tends to, you know, but this thing about, you know, how do you reform regulations in a place where the regulations are not well known anyway. And it is, you know, I think one of our key findings will be is, you know, how do you know, trying to find these different ways that people do things that are, and I think similar to what Claes said, or, you know, what people are saying, it's not just about, you know, we're all looking at government reform here, where actually it might be the multinationals you need to look at. There might also be, you know, a space where you have to start looking at the more, what are classed as informal ways that people are doing this. Yeah, and I don't know if Jose, because Jose is sort of much more closer in the sort of policy level. If you want
yeah, well, I think you just made some really good points, and there is not much to add, but I think it's interesting to think about that contrast between, like, I mean, in certain settings, you just have to work with what is available. And I thought a lot about it when Steve was talking about this problem of having tested for the presence of E. coli and that leading to the direct use of certain antibiotics instead of and then trying to reivindicate or emphasize the need to understand the issue in context instead of just taking the positive test as an indication that antibiotics should be used. But then we come from the kind of context that we were looking at. Like, that test is not possible, it's not feasible, it's not accessible economically. So you just have they actually have to work with that kind of sensing of whether there is disease or not. So they are forced to work without the test. So I think in the kind of setting that Steve was describing, there is a little bit of flexibility to. There are different elements that you can combine to try and improve the way people use antibiotics. But in the setting that we were explaining, there is absolutely no flexibility. I mean, what we have is what we have, and that's exclusively the experience, the kind of reading that the breeders and the veterinarians can do with the tools that they have, which are very limited. So even if you bring the regulations, there is no flexibility for the regulations to work in any way. It's so the situation is so delicate and so balanced that any kind of movement might, push it over towards losing your livelihood for that month, or even for a longer period.
Thanks, Jose. I mean, I think you're echoing again we were talking at the beginning, Steve started out with this kind of tension perhaps between the ideas of individual choice and the ideas that there are structures and that there are other things in the middle or muddle. And I think what we've seen across here, there are all of these devices and tools and so on, and this muddle that we're in as social scientists of grappling all of these issues. And so the slightly the interesting point that Ridgell kind of brought in, is the antibiotic era merely a blip, and the exception to the rule kind of makes us, see this different horizon of what we're doing. And I think this, brings in the challenge in quite a salient way. So we're coming to the end of our session. So I would like to say a thank you to all of our panelists. This is really interesting. We have not arrived at the solution or the conclusion, unsurprisingly, sorry, but we have, I think, got a bit further, because I think listening to these different perspectives does push us towards some additional questions and ways to ask questions that perhaps can help us to move in a useful and productive direction. I'm just going to flash up very quickly the next the slides. I'm sorry we haven't answered all of the questions that were there online, but I hope you agree we had an interesting discussion nonetheless. We have the upcoming panels are, the next and final panel for the season is on 17th of December, which is on antibiotic science, technology and infrastructures. And we're very fortunate to have Catherine Will, Kamatra Chinsatyansup, and Charlotte Breves and Nick Brown all giving us talks on the 17th of December. And then in February, we have a different kind of event that will be different kinds of panels, not just for social scientists, but for us as social scientists collectively, to put forward our ideas that culminate from across this series of panels, and to hear what other people outside of the social scientists make of those. So so we'll be sending out information about that for all those who have clicked on yes in the poll, and you'll be able to find us from there. Or follow the Twitter from any of these Twitters. So just to say thank you very much again to our panel. Thank you everybody for joining us. It's been another great one, and enjoy the rest of your days, evenings, mornings, wherever you are in the world. Thank you.